Hello guys again. Today we'll talk about the citric acid cycle and oxidative phosphorylation to produce the energy from glucose. We did last time about glycolysis, and in glycolysis we are using one molecule of glucose to produce two molecules of pyruvic acid. That two molecules of pyruvate acid or pyruvic acid, they are 3 carbon while glucose is 6 carbon. So, and the net production from this glycolysis about the energy, we are producing net 2 ATP and you are producing net 2 coenzyme reduced NADH. So here is the net production of energy from glycolysis, and here the production from pyruvate. This happens in systole. Then the pyruvate are traveling from the systole to the matrix of mitochondria, and while it's transporting to the matrix of mitochondria, pyruvate are converted to something called acetyl coenzyme E. That process, you can see here in this red color, is called transition state or pyruvate oxidation. Exactly what happened is oxidation decarboxylation. For example, here is the most important enzyme we did last time, is called pyruvate dehydrogenase complex. And you are producing here CO2, and we reduced the coenzyme NAD into NADH. So here is the production and acetyl coenzyme E. Of course, you need to insert coenzyme E here. So now we are inside the matrix of mitochondria. So what happened? What is the steps behind this one? Just to make it clear, here is, mitochondria has outer membrane and it has also inner membrane, which is folded like this one. And you can see, guys, that we have kind of transporter founded here. That transporter will bring the pyruvate. Pyruvate, how many carbon, guys? You remember? It is 3 carbon. So we have here our pyruvate, and we have 2 molecules. So we have 3 carbon molecule, and we have from that one 2 pyruvate. This one are transporting inside the matrix to convert to, you remember, transition step. This one will convert it to a specific molecule called acetyl coenzyme A after we losing CO2. So we have here one molecule which is 2 carbon, but that molecule is bound to specific structure called CoA. So we have this structure here is called acetyl coenzyme E. That acetyl coenzyme E will have a condensation reaction for the other 4 carbon molecules. So we have other 4 carbon molecule like this 1, 2, 3, 4. That carbon molecule is, we call it oxaloacetate. So this acetyl coenzyme E with the oxaloacetate, both of them come together in reaction called the condensation by a specific enzyme. That enzyme is called the citrate synthase. So we have here citrate synthase. That enzyme, which can make this condensation, will produce 6 carbon molecules. So we have here kind of 6 carbon molecule, 2, 3, 4, 5, 6, and there is no coenzyme. So you can understand within that reaction, we are bring the coenzyme outside. We are bringing the coenzyme outside here. So here the coenzyme are going. So we lost the coenzyme here. That 6 carbon, the first 6 carbon produced in the citric acid cycle is called citrate. So we have here citrate. That citrate is 6 carbon. For that we call the cycle citric acid cycle because the first molecule are produced is citrate. Now the point is, you have acetyl coenzyme A coming from pyruvate condensate with the oxaloacetate and producing citrate, which is 6 carbon. Look guys, this reaction we have kind of intermediate are formed. يعني before we have a citrate we have a specific intermediate found here. This called cytoyl CoA. يعني before citrate we have something called cytoyl CoA. That cytoyl CoA, it is very fast to hydrolyze. So when this is hydrolyzed immediately, so it's not staying for long time. This will make the reaction irreversible. So that reaction is only in one direction, not in two directions. Remember it, very important to remember it is irreversible. Once the reaction is irreversible, it means it is very high regulated reaction. Very high regulated. How this reaction is regulated? Simple way that I will say here what factor can activate citrate synthase, inhibit citrate synthase, and what factor activate it. Logic say these reactions are one of the reactions to produce energy. So my target is producing energy. So you think if you have enough energy molecules, you should make it or no? Of course no. If I have enough ATP, why I should make citric acid cycle and oxidative phosphorylation to produce more ATP? So we can expect that increase amount of ATP will inhibit it, increase amount of NADH inhibit it, and the increase amount of citrate itself. يعني citrate itself, yes, like working by negative feedback, allosteric regulation. يعني citrate itself as a product of that enzyme can inhibit the enzyme if I have enough citrate. And also one of the other molecule which formed later in citric acid cycle is called succinyl coenzyme E. So here is the factor which inhibits citrate synthase, which inhibits citric acid cycle, while it can be activated by ADP. So ADP is the factor which activates the enzyme, while the other factor inhibits the other enzyme. The other factor inhibits this enzyme. So we're reducing citrate now, and you can remember something. Remaining reactions, total reaction in citric acid cycle now. What is the main target? After citrate, I will have 2 carbon released as CO2. يعني from the total reaction here, if I need to make a cycle like this one, I will convert the citrate to oxaloacetate. يعني the result is citrate will become oxaloacetate. Oxaloacetate is 4 carbon, citrate is 6 carbon. So I will lose 2 CO2 from one citrate. Okay guys, and you remember something also here. Oxygen is very important for that reaction, but not for the reaction itself. I will explain this for you, but remember, citric acid cycle, oxygen required. So whenever we make citric acid cycle without oxygen, I will explain this. Let's go for the steps and I will explain why the oxygen is very important. Make this question in your note and ask yourself at the end of lecture why we need oxygen here. Okay, so after citrate are produced, we have this kind of dehydration and dehydration. Yani we have a reaction which is called dehydration and dehydration. What does mean dehydration? Dehydration means I release water, and dehydration means I add water. So we have the two reactions happen together. Who can do this two reaction, guys? A specific enzyme called aconitase. Aconitase. So we have here that aconitase enzyme. It is the enzyme first to make dehydrate and then hydrate citrate. So there is no change in the carbon number of the carbon, still 6 carbon molecule, and that 6 carbon molecule kind of different arrangement. Okay, so the same number of carbon, but we change the structure of the distribution of atoms. So we have again, it is 1, 2, 3, 4, and just to make different color, to just remind you, this blue one, it is the acetyl, and this red one, it is the oxaloacetate. So the number of carbon is still the same, but what we only change the distribution of the molecule, distribution of the atoms within the molecule. Okay, this 6 carbon one is called isocitrate. So here we're talking about isocitrate, and it is 6 carbon molecule. Okay, after isocitrate, we will find, and by the way, this reaction is reversible reaction. So remember, it is reversible reaction. It is very important to know which reaction are reversible and which reaction are irreversible. So isocitrate will be oxidized and decarboxylate. Look for the expression, oxidized and decarboxylate. So let's make this arrow like this one. And you say to us that is it oxidized? Yes. How? By dehydrogenase. What does mean dehydrogenase? I will get hydrogen atom out. So who can carry the hydrogen? Who can carry the proton? Coenzyme. What coenzyme need here? NAD. So we have here NAD will be reduced to NADH. So here is the dehydrogenation. Okay, so what is the other step? I also will bring out CO2. So it is the first CO2 produced in citric acid cycle. So this reaction is called what? It's called oxidative decarboxylation. So here, oxidative decarboxylation. I write it here. Oxidative decarboxylation. Okay. This oxidative decarboxylation will produce what, guys? For isocitrate, will produce a specific molecule. You think how many carbon now? That molecule is how many carbon, guys? That molecule is 5 carbon. So I'm losing 1 carbon. So I will have 1, 2, 3, 4, 5. So here, this is called alpha ketoglutarate. Alpha ketoglutarate. Okay, so now we have 5 carbon molecule alpha ketoglutarate. Don't forget. And what is the enzyme who can manage it? This reaction is irreversible. And what kind of enzyme can regulate it? It is regulated by isocitrate dehydrogenase. So the enzyme here is called isocitrate dehydrogenase. Here is the enzyme who manages this step. Please remember it. Isocitrate dehydrogenase, yes, that enzyme is highly regulated. What factor inhibited and what factor activated? It's the same concept. It's inhibited by what? Inhibited by energy. So ATP will inhibit it. And what factor activated? Of course, it is ADP. Plus calcium. Calcium here is very important. Like skeletal muscle need to contract. When the skeletal muscle going to be contracted, what factor can help us? Help it to be contracted? Calcium. So the calcium increase in the muscle which is highly contracted, and that calcium also will help to produce ATP which is needed to make the muscle contracted as well. So the calcium is playing here many roles. If we study the contraction of muscle, we need calcium to make the cross bridge between actin and myosin, and at the same time we need the calcium to increase the number of ATP produced. So this one is activated by ADP and the calcium, and inhibited by ATP and even NADH. The same logic يعني. The same logic. Okay. After we have this 5 carbon molecules called the alpha ketoglutarate. Alpha ketoglutarate. We'll make again something called to produce from alpha ketoglutarate succinyl coenzyme A. From alpha ketoglutarate to succinyl coenzyme A, again we will make oxidative decarboxylation. Oxidative decarboxylation, yes. So we need here what? We need that alpha ketoglutarate to convert to 4 carbon molecule. What 4 carbon molecule? It means what? It means that I will produce CO2. So we have here CO2 produced. And it is dehydrogenase. Yes, dehydrogenate means oxidative decarboxylation. So what does mean oxidative decarboxylation? Decarboxylation producing CO2. Oxidative means dehydrogenation. So NAD coming as oxidized to be reduced to NADH. Okay, so now we have one molecule. That molecule what we're producing, it is how many carbon, guys? It is 4 carbon. 4 carbon, like this one. Okay, so we have here 4 carbon molecule. What is the name of that 4 carbon? But just a moment. We make this one, and if I'm telling you that during that step I insert here coenzyme A, yani again I lost one coenzyme A here and I bring it here. So I have coenzyme A inserted in that reaction. That coenzyme A will be added to the carbon number 1, by the way. Here is coenzyme A. So what do you think, guys, this one? What do you think? That is the molecule called the succinyl coenzyme A. So we're talking about succinyl coenzyme A. That succinyl coenzyme A is kind of oxidized the form of alpha ketoglutarate. I'm using different expression because this is the questions. So alpha ketoglutarate is oxidized to succinyl coenzyme A. What kind of oxidation? By dehydrogenation and also decarboxylation. So I'm producing CO2 and I'm producing NADH and I add the coenzyme A. What is the enzyme who can manage this step? Just like me, let me do this is better way. Here we are insert here the coenzyme A, and I need to write the name of the enzyme here who manages this one. It's called the one who make dehydrogenation for alpha ketoglutarate. Look, guys, yani just to help you, isocitrate dehydrogenase to convert isocitrate to alpha ketoglutarate. Then I do dehydrogenase for alpha ketoglutarate to make succinyl. So it's called what? Alpha ketoglutarate dehydrogenase. So that enzyme is called alpha ketoglutarate dehydrogenase. This is the enzyme which is responsible to oxidize alpha ketoglutarate to succinyl coenzyme A. Again, we need to know what factor activated, what factor inhibited. Don't forget what factor inhibit and what factor activate. The factor which inhibit, again, it is the energy, especially what? NADH and succinyl coenzyme A. So we're talking here NADH and the succinyl coenzyme A. And it's activated by calcium. This one activated by calcium. Activated by calcium. Very important. Alpha ketoglutarate. This one, alpha ketoglutarate, is looking like pyruvate dehydrogenase, what we studied together. This one looking like pyruvate dehydrogenase. Pyruvate dehydrogenase, what we studied before. So here, alpha ketoglutarate dehydrogenase to looking like pyruvate dehydrogenase, but it's act on alpha ketoglutarate and they produce succinyl instead acetyl coenzyme A. Am I clear, guys? Okay, but the coenzyme of these two enzymes are identical, like NAD, H, and so on. Okay, so now we have succinyl coenzyme A. After we have succinyl coenzyme A, we need to convert it to other molecule only by change in the atom distribution again, and losing the coenzyme A. This is from succinyl coenzyme A, I converted to succinate. So here again we have still 4 carbon molecule, but there is no coenzyme A, so directly you expect that we kicked out the coenzyme A here, so don't have it. But we have one step here called substrate level phosphorylation. What does mean? Phosphorylation means you add a phosphate to ADP, but we have two systems to add a phosphate to the ADP. Maybe we make a phosphorylation for ADP in the matrix of mitochondria after electron transport chain. This is called the oxidative phosphorylation. And maybe we can produce ATP by getting the phosphate group from specific molecules and add it for a specific ADP to produce ATP. This is called what? Substrate level phosphorylation. Remember it. So we do what here? We do substrate level phosphorylation. How? We have a specific molecule called GDP, which will be phosphorylated to produce GTP. GTP is looking like ATP. So GTP is looking like ATP. So GTP will bind with ADP to convert it to ATP. So we can say now, simply we're producing one ATP. Simply. Succinyl coenzyme A is converted to succinate. Converted to succinate with the formation of one GTP and release of coenzyme A, as you remember here, release this coenzyme A and producing GTP. You know, the enzyme which is responsible for that step is called succinyl coenzyme A synthetase or thiokinase. Yani we can say that enzyme is called thiokinase. Succinyl coenzyme A, succinyl, or we can say succinyl thiokinase. Okay, this thiokinase, this one, where GTP are formed, the only place where GTP are formed here. We don't have other place in citric acid to produce this one. Okay, so now we have succinate. Succinate is 4 carbon as well, 4 carbon, and here is no dehydrogenation. I remember it. After succinate are produced, we need to make dehydration for succinate. So succinate dehydrated means I'm losing hydrogen. So let me write it here. So we have a specific enzyme here. That is specific enzyme which will produce, again, you remember, we have NAD, not NAD now. From succinate to be dehydrate, we have 4 carbon to form one molecule called fumarate. It is kind of dehydration, so I'm not using NAD, I'm using other coenzyme called FAD. That FAD is reduced to FAD H2. FAD has more affinity to hydrogen than NAD. For that, if you're using NAD here, that reaction will be reversible to the direction of succinate. Would be irreversible in the direction of succinate formation. But we have FAD to make it reversible. So the FAD will make this reaction reversible. And producing FAD H2. The enzyme which can do this one, it is very specific enzyme. It's called, again, succinate dehydrogenase, and this is the complex number 2. يعني this enzyme found in the inner membrane of mitochondria, found in the membrane bound enzyme. This enzyme is a complex number 2 into the electron transport chain, So it's very important to remember this one. Succinate are dehydrogenated or oxidation to fumarate, and this succinate oxidized to fumarate by a specific enzyme called succinate dehydrogenate, which is found in complex number 2 in the inner membrane of mitochondria. So we have fumarate now, producing fumarate. Then after you have a fumarate, you can make something called hydration. Hydration for fumarate, what does mean hydration? Hydration for fumarate means we add water for this one. So here, remember, we insert one water here. So what happened now? Again, it is kind of change in the because fumarate has a double bond between the carbon number 2 and number 3. When we add hydrogen, there is no double bond. I think that chemical details, what happened between the atoms, is not important for you, but just know, when we add a water to make a hydration, this is kind of change in the structure, because between the carbon number 2 and the carbon number 3 was there double bond here. After you add water, there is no double bond. Here we have 1, 2, we have 4 carbon from here. After we have succinyl, and the last CO2 are loosed from alpha ketoglutarate. Succinyl, everyone is 4 carbon, and just we make a change in the structure of this one. And this is reversible, and the enzyme which do this one is called the fumarase. So here is the enzyme, is called fumarase. So we have fumarase enzyme which make hydration for fumarate to produce a specific enzyme called malate. And from malate, which is 4 carbon, will convert it to the oxaloacetate. So malate convert to oxaloacetate. And please remember, here we're producing also NAD. So we have here NAD will reduce to NAD H. So producing, or we make a oxidation for malate. That oxidation for malate will result in what? Producing oxaloacetate. So what the enzyme, we should remember the enzyme here is called malate dehydrogenase. This is very important enzyme. This looking like lactate dehydrogenase. It's called malate dehydrogenase. So malate converted to oxaloacetate by that enzyme, and we are producing one NADH. The malate dehydrogenase looking like lactate dehydrogenase, and this one could be irreversible if we have low NAD. Look for this one. Yani that reaction will be irreversible if we have low NAD, but we are inside the matrix, so we have more NAD. This will make the reaction is reversible. So I will highlight for you here how many reactions are irreversible, and they are very important for us. Please remember them. We have citrate synthase, it is irreversible enzyme, very important. And we have from isocitrate to alpha ketoglutarate, it is irreversible, very important. And from alpha ketoglutarate to succinyl coenzyme A, it is irreversible, while the other reaction, all of them are reversible. And please remember, the enzymes which working for irreversible directions, they are highly regulated. So for that also, I will highlight for you that enzyme which is highly regulated, like citrate synthase is regulated by ATP NADH citrate succinyl coenzyme A will inhibit this one while ADP will activate it. And the other enzyme, it is isocitrate dehydrogenase. It is very important. And the last one, alpha ketoglutarate dehydrogenase, and also is very important. Here is the steps for citric acid cycle. Then we should ask ourselves what kind of energy produced from there, how many energy you are produced from there. Look guys, let's count. You talk here about one pyruvate and one acetyl coenzyme A. So let's count the number of energy produced from one acetyl coenzyme A. I also will highlight this one because you need to know from where, from which step we're producing. Look guys, we produce this one. First one we are producing as an energy, it is kind of NADH producing from isocitrate dehydrogenate to alpha ketoglutarate, and one NADH also from alpha ketoglutarate to succinyl coenzyme A, and NADH from malate to oxaloacetate. So we have like 3 NADH. Okay, do we have something else? Yes, please. Count with me. How many FADH2? FADH2 is coming from succinate to fumarate. NADH2. FADH2. How many? 1. I'm talking about the produced from one acetyl coenzyme E. Okay, and how many ATP? We're producing one ATP. One ATP. Where is it? From can you see from succinyl coenzyme A to succinate we're producing 1 ATP. Okay, how many CO2? We're producing 2 CO2. From where guys? We have 2 steps, from isocitrate to alpha ketoglutarate, and from alpha ketoglutarate to succinyl we have 2 CO2. And how many coenzyme A? We're producing 1 coenzyme A from here. Let me get it. 1 coenzyme A I produced here, and I will produce 1 coenzyme A. So we have producing also 2 coenzyme A. Here is the total production from 1 acetyl. But if you talk about the production from, remember the production from 2 acetyl, because we have 2 pyruvate, 2 acetyl, so you can make this number times 2. So here is times 2. So here is very important. So mainly, or you can make it easier for you, I just change the number. The 3 will become 6 and 2, 2, 4, 4. So we can say 6 NADH, 2 FADH2, and 2 ATP, and 4 CO2, and 4 coenzyme A. Total production from 2 molecule of pyruvate. So here is the produced things from the citric acid cycle. There is one question we didn't cover yet. Why citric acid cycle oxygen required while there is no one step you told us that there is oxygen needed here? So the question answer is like this guys. Please remember, if you don't have NAD, can you make this reaction? No. If you don't have NAD, can you make this reaction? No. If you don't have FAD, can you make this reaction? No. So what happened now? I need the oxygen to eliminate the proton from the coenzyme reduced form. So I need to make this NADH to be NAD again to continue the cycle. So I'm going to explain to you now how we can make the NAD to continue the citric acid cycle, because without NAD, without oxidize the form of that coenzyme, we cannot continue citric acid cycle. So the main target for us for the coming slide is how this NADH is oxidized to NAD, how this FADH2 oxidized to FAD, and here is the electron transport chain.